Working with hepatitis viruses for the last 40 years, mainly in developing vaccines. And through this interest, at one point of time, I met a number of colleagues whom I'm going to talk to you about soon, who actually triggered my curiosity, and I got into this area, which was completely new for me, but is really fascinating. Evolution of this study started actually some years ago when I got a phone call, just to say that I was quite hesitant whether to go into it, but you will see from the results that this is a whole new world that has started, and I'm going to describe it to you in a few words. I'm going to talk to you about the hepatitis B virus. It's a DNA virus. It is about 3,400 base pairs long. It's a partially double-stranded DNA virus, and is well described, and the most, most samples of the hepatitis B virus, or most isolates, were obtained since the 1950s, not before. So we didn't have any information on the age of the virus, how ancient it is, and practically all isolates that were available start from the 1950s, more or less. What is so special about the hepatitis B virus? That the virus infects the liver. It is hepatotropic. It homes to the liver and hides in the nucleus of the virus, of the hepatocyte, of the liver cell, in a molecular form that we call cccDNA, covalently closed, circular covalently closed DNA. And it hides there even in patients who recovered from hepatitis B. And most patients who get acute hepatitis, get infected, they clear the virus if they are not infected around birth. Because when they are infected from their carrier mothers, this is a different story. Their immune system becomes tolerant to the virus, and then they become carriers, the newborns and the babies. But adults, or even young adults, when they get infected, clear the virus, but the virus continues to hide in this cccDNA form in the nucleus and stays there forever. Now, what is so important about this virus is that about 400 million people are infected worldwide and carry the virus, mainly in Africa and in Asia, but today also in many other countries, as you will see. And about 10% of those infected become carriers, and consequently some of them will develop what we call chronic hepatitis or cirrhosis, scarring of the liver, and about 5%, an annual incidence of 5%, they develop a malignant transformation and have liver cancer after the age of 50 or 60. So it is in a disease which affects millions and millions of people. In China alone, there are about 150 million carriers of hepatitis B. And this is true in many countries in Africa and in East Asia. Now, when we look and screen for hepatitis B in the world population, we find that about 2 billion people have been infected with hepatitis B at this point of time. Most of them recovered, or so-called recovered, but the cccDNA is still in the nucleus, in the hepatocyte. And what is also not less important, that some of this DNA integrates into the host DNA. Now, this disease was really a very significant threat until about the late 1970s, when the first vaccine was developed from viral proteins isolated from carriers of the virus. Today we have recombinant vaccines which are outstanding, and about 170 countries today immunize all their newborns against hepatitis B. So the problem for those who are born now is really not very serious, but we still remain with this enormous amount of patients worldwide, and we do not have a very efficient antiviral agent to clear the virus, although there are much better methods today to treat those patients. Now we divide the world according to the prevalence of the viral surface protein. We call it hepatitis B surface antigen. Here, so this is the envelope protein, which is thought for in all blood banks. Before the blood is given, we screen for the hepatitis B surface antigen, and we divide the world into high, intermediate, and low endemicity according to the prevalence of this surface antigen. Now, I will not go into details about the origin of viruses, but as you know, the origin of ancient viral genomes is a matter of speculation, because there are very few viruses which have really been sequenced and cloned. And the comparative sequence analysis suggests that DNA viruses have an archaic evolutionary roots what concerns their organization, and there are certain domains, genomic domains, which can be followed if you have the right sample. And there are quite a number of mechanisms where DNA and RNA viruses are really generated, and they usually are characterized by mutations, recombination, and reassortment. Many of you are familiar with the fact that, for instance, RNA viruses have a very high mutation rate, like the HIV virus or the hepatitis C virus, but this is also true for the hepatitis B virus. Now, all these substitutions may have very practical implications. So if we could forecast these changes, we could also design better antiviral agents or better vaccines and avoid drug resistance and eversion from vaccine escape mutants. Saying all this, I'd like to talk about the hepatitis B virus, which belongs to the HepaDNA virus family. And as you can see, this is a quite large family. I won't go into details here. Every year we hear about new members of this group, and apparently this is a very, very old virus. And what is important is it's not only affecting humans, but also it affects primates. And as you will see, some of the theories about the evolution of hepatitis B come from primate studies, which have been used for years as a model to study the clinical and the molecular pathophysiology of hepatitis B. But in the last 10-15 years, there is a ban on work with primates, and we hear less and less about it. But especially chimpanzees were used to develop the new vaccines against hepatitis B. Chimpanzees do not get sick, they don't get liver cancer, but they become carriers when they are infected. And probably they have infected their handlers for years before. There are also models in ground squirrels, in woodchucks, in bats, of viruses which have a very similar sequence of hepatitis to the hepatitis B virus. Now, what are the factors with potential impact on the diversity during evolution? First of all, there is an interaction between the host and the pathogen through immune response, and also following vaccination. By vaccination, we create an immune pressure, which can lead also to escape mutants, and this has an implication on what I'm going to show you soon. There are, of course, DNA recombinations, and the antiviral treatments that we are giving today causes selection of mutants sometimes, which are very difficult to handle. So there are lots of theories how the hepatitis B virus has evolved, and very little proof. I mean, there is an investigator by the name of McDonald who swears that this is the hepatitis B virus is of primate origin, starting in chimps and woolly monkeys and orangutans, and co-involved with the primate hosts for a period of 10 to 35 million years. And then the cross-species contamination occurred in Africa, and from there it was passed to humans. Helen Norder in Sweden thought that the HepaDNA viruses have a common ancestor to men and apes, and she described two lineages. I will not go into detail, but she is one of the forefathers of the investigation into the origin of the investigation of HBV. And then there is a new world origin of HBV theory that is spreading from the Americas over the last 400 years, following cross-species transmission from New World monkeys to humans about 2,000 years ago. And finally, recently, there is a paper that suggests that the hepatitis B virus jumped, quote unquote, into humans about 33,000 years ago. Now, when we look at the virus, there are at least 8, probably 10 genotypes and a number of subgenotypes of the virus. The genotypes are defined by an 8% divergence of sequences, and the subgenotypes about 4 to 8%. And in fact, today, when we investigate outbreaks, we actually study the molecular epidemiology of the virus. And if we have a spread, for instance, in a hospital, an infected healthcare worker is infected, if we look for the source, we can actually identify the genotype and the subgenotype and trace the origin of this isolate. So the world is divided into areas where certain genotypes from A to F are spread, and you can see that in some countries there is only one genotype, mainly one genotype, like in South America, genotype F, but in the majority of the countries in the world there are mixed genotypes. Now, from the 8 genotypes, from A to H, there is one genotype called genotype C, which is very prevalent in Asia. And the question is always, how did this genotype C got into Asia? And how? Where does it come from? And from the 8 genotypes, each genotype has its own character. For instance, genotype C is more frequently associated with severe liver damage and liver cancer, while genotype B, for instance, is associated with less active disease, slower progression, and lower incidence of liver cancer. And if we go further, genotype A and B have a higher rate of response to treatment. So there are differences which are sequence-based of the various genotypes. Now, genotype F, which is mainly in South America, is characterized by the tendency to destroy the liver. So there are many more cases of fulminant hepatitis which require liver transplantation in South America as compared, for instance, to the Mediterranean area. Now, this would be very simple if we would have these isolates which are very clear, and the world could be divided into the molecular epidemiology of the genotypes. But today we have waves of migration, and the migration also brings the various moves the various genotypes from one part of the continent, one continent to another, and consequently there is total chaos in the molecular epidemiology of hepatitis B, because we can find most genotypes in most places. And if we want to go into the study also about the origin of hepatitis B, we have to look where there is a region where there is a homogeneous population of virus in this particular area. This is the reason we went to Korea, actually, and this is all by chance, and I will then pay the tributes and acknowledge to all these investigators who really made it possible. Just to say that most of the molecular work was done by Gila. Where is Gila? Here she is. So she did most of the work, and we were more in the background. And the reason we went to Korea can be seen here in this slide that is coming, that you can see that from all countries that we have investigated, Korea is characterized that 100% of the hepatitis B virus infected individuals are of genotype C. So it is a very, very homogeneous population. If you look elsewhere in East Asia, you see there are many mixed populations in the other countries. Now, when you look at South Korea, these are the sites where mummies have been excavated in various places in Korea. There has been an enormous wave of construction in Korea, where old graveyards, which are sometimes family graveyards, have been moved and excavated to make some space for high rises and new buildings. And consequently, in Korea, many of such graves and mummies, because they have been mumifying their dead for a long period, had to be moved. And this created the opportunity for our colleagues in Korea, whom I'm going to mention later, to obtain some tissues, hopefully with some hepatitis B virus, but we didn't know this in the beginning. Now the mummification technique in Korea, I'm not very familiar with it, but I understand that most of these mummies underwent natural mummification, and they have apparently some special embalmment techniques in some places, but most of them underwent natural mummification. One of the major problems when you look for livers in mummies is that you don't find them, because the Egyptians, for instance, used to put them into jars and they rotted there, and you cannot really get parenchymatous tissues, if it is liver or spleen or whatever, because it's unavailable. And there is I found only one example from Peter Scheuer, who was a late British liver pathologist, who, I don't know where he got this sample, but he presented it years ago, maybe 25 or 30 years ago, as a histology of a mummified liver. I've never seen, I don't know where it comes from, but one of my friends who heard my lecture sent it to me, and unfortunately Peter Scheuer is not with us anymore, but this is a slide from his collection. In Korea, apparently, the mummies have been put into in caskets, and they were sealed. And probably in this technology, some of these parenchymatous organs were preserved. And actually, almost 2/3 of the bodies are naturally mummified. The temperature fluctuates, but it's about 10%. And the coffins are of pine, and probably the way these mummies were preserved also contributed to the preservation of the DNA. This is a photo which was published by our Korean colleagues of one of the sites where our mummy was excavated, and you see how it looked for the first time when it was there. And this is also the design of the casket where the mummies were put in and preserved over the years. And by the way, I just want to say that since we are physicians, before we started this work, we asked for permission of what we call the Institutional Review Board, the IRB. You know, you cannot do clinical trials today without the approval of an IRB. But how do you get approval of an IRB for a mummy? That's not so simple. So where I actually refused to start working before I got permission from the university in Korea, and they had a big debate, but finally they gave us a consent. Apparently, we were the first who were asking for it. In the past, they never thought about it. The focus of my presentation is indeed this particular mummy, which is a 4 to 6 year old child that was excavated at the site I've shown you, and this is how it looked after excavation. What is more important is that before our Korean colleagues started to work on the mummy, they have done a CT scan of the mummy, and as you can see from this CT, they identified a parenchymatous organ in the right upper quadrant of the abdomen. This is the area of the liver, and this is the area of the spleen. So this is the site they have seen, and they actually biopsied this liver through laparoscopy. As I showed you before, they came from the back, and this is how they did it. You know, they did a laparoscopy like they do every day in a laparoscopy there, and got quite a number of liver biopsy samples weighing about 1 gram per sample that have been sent for analysis. Before the project was started, all investigators and all physicians and colleagues in Korea were tested for hepatitis B, because the issue of contamination is, of course, very, very significant. And just to remind you that about 15% of the Korean population are carriers of hepatitis B. 1/5. This is unbelievable. 15%. So we had to be very careful that, first of all, those investigators are immunized who are not carriers, and investigators who are carriers could not participate. Now, one thing which I will come back to you later is this is the site of the laparoscopy. This is the liver, as you can see. It has a leathery appearance, and this is the abdominal wall, but the inside, the visceral part of the abdominal wall. And I will come back to this a little later. We have obtained some tissues and did some hematoxylin eosin staining in the laboratory here at Hadassah, and you can see that you cannot find any liver cells there anymore, really, at least not by microscope, but you can see fibrous tissue. The blue staining is the staining of fibrous tissue, which is usually a reaction to an inflammatory process in the liver. But we couldn't, of course, identify liver cells. The first question was, how old is this mummy and how old is this liver tissue? And C14 radiocarbon dating was done in Korea and here in Israel at the Weizmann Institute, first of all on clothes that was found in the coffin of the mummy. And you can see that the silk was dated back to the 1530s, the cotton to 1360, and the liver about 1680. One could ask that how do these match one to another, but first of all, carbon dating is not an absolute exact science, and the second thing is that apparently clothes at this time were passed from generation to generation. So they were not necessarily new. Our Korean colleagues did one try to look for hepatitis B and found a PCR product of about 239 base pairs, but they were not sure, and they asked actually us and another laboratory in the University College of London to confirm the presence of the hepatitis B virus DNA. And this was the result, and all three laboratories confirmed the presence of a small band of HBV, 239 base pairs out of 3,400 base pairs. I don't have to tell this audience about the pitfalls of sequencing of ancient DNA, I understand, so I will skip these slides which I usually show in the university. And just to say that we took 10 milligram biopsy samples incubated with bleach because we were not sure regarding contamination, and then did the DNA extraction as usual, genome amplification of about 44 overlapping primer sets, and the PCR products were separated on an agarose gel, and the positive amplification products were purified, directly sequenced, or cloned and sequenced. What the most important findings were that the ancient HBV DNA consensus sequence was identified, and it clusters with a published hepatitis B genotype C subtype 2 from Korea, Japan, and China, and differ from hepatitis B C1 sequences from Thailand, Vietnam, and Myanmar. All these comparisons were done with samples which are relatively new. About 160 ancient DNA sequences differed among themselves by 4-point mutations, as is shown here, and the ancient HBV amino acid sequences clustered together with other published hepatitis B C2 genomes and are quite similar. About 18 amino acid sequences were found in the samples that we have tested. Actually, Gila did. She did not find any recombination events along the ancient DNA suggesting establishment of an HBV DNA from a common ancestor in China or in Japan. As you can see, this is a map of the 44 short HBV DNA overlapping fragments, a work which took some time, but actually this is the most ancient hepatitis B virus ever identified, and so far there are no other samples in the world. And this was of course deposited in the gene bank and is available for comparison, and the phylogenetic placement and historical context of it was published in the Journal of Hepatology, and you can look at it here. And we can actually put the ancient DNA sequences from Korea in context with the other sequences. Here it is shown in black to show how it fits into the dendrogram here. So the main findings and conclusions: this isolate most likely represents the earliest human hepatitis B sequence that colonized Southeast Asia by human migration from a mummy that we identified in Korea. An analysis of the complete ancient DNA genome stitched together from 24 separate liver biopsies revealed a unique HBV genotype C2 sequence commonly spread in Southeast Asia, most likely representing an HBV which infected the Joseon dynasty in Korea starting on the 14th century. In comparison of the ancient hepatitis B sequences with contemporary sequences revealed distinctive differences along 4 open reading frames, and the calculated divergent times is shown here. I won't go into more detail since my time is out, but I would say that as I started this presentation, you can see here today that if we wouldn't have gone to Korea or to other places, it would be more difficult to find really samples representative of a certain genotype. You see, genotype C started to migrate from East Asia over Russia to the Bering Straits and then moved into North America and South America. And this is true now for immigration from Africa to Europe and so on and so forth. So today the world is full of immigrants from all these areas who have brought with them the genotype of their particular country, and in Europe alone there are millions of carriers of hepatitis B from Africa and Asia. To finalize, I just want to show you the abdominal wall of the laparoscopy of this Korean mummy. because we knew that hepatitis B is not a killer in young children. They usually are silent carriers. And we looked at these nodules, and it is highly suggestive that these nodules are actually tuberculous nodules. I mean, we have, I'm not free to show you all the data because we didn't do these experiments, but apparently there is already a positive PCR in situ hybridization from a group in the United Kingdom. And probably this is the reason why this child died and not hepatitis B. And in addition, you can see these nodules here. This is highly suggestive of peritonitis due to tuberculosis. Here you can see the nodules again. And finally, the last slide, you can see that there were quite a number of parasites in the stool of this young man, and these included the eggs Clonochis siensis, Ascaris, and Trichuris trichura. Finally, last but not least, to mention the co-workers who did this work. This is not a one-man show, of course. I just want to mention Mark Spiegelman, who was a matchmaker, that he spent 3 years of diplomatic interaction with our Korean colleagues, where there was a serious language barrier. This was not easy, but he finally managed to do it with hundreds of phone calls and interactions. And of course, Gila, who was a driving force in investigating these tissues. These are our Korean colleagues mentioned here by name. And I also would like to mention Atalia Klein, the third on the row from the left, who was our technician in our laboratory here in Jerusalem, who had also a major input in this study. Thank you very much.
Questions? So I have a question concerning the dating. So you kind of skipped that, unfortunately, that slide. I was curious about, so you calculated the most recent common ancestor for the lineage C?
Yes.
So I'm just curious about viruses having kind of really different mutation rates, I guess. So it must be quite, especially with such a small genome, to really then calculate the mutation rate based on something that evolves really fast with very different
I must say that the hepatitis B virus is remarkably stable. I mean, the changes compared I mean, the state today compared to what has now been shown in the 16th or 17th century are minimal. So it is relatively stable. But maybe, Gila, you would like to comment on it.
Well, there's some data about contemporary viruses, the hepatitis B. So we took some of their estimates of the change. But,
okay.
So
you didn't use it as a tip calibration point, the ancient genome, say with the radiocarbon
rate.
rate.
So we tried the various stages. We dated it and we took different estimates for the and I think one of the interesting thing about this sequence is that this is pre-pharmaceutical use. So I think then what you have is a lot of changes that you got due to the adaptation to different medication that was given.
Still incredibly still kind of long branch, if you want, given this time period, 300, 400 years. I would think like virus evolution would be much faster.
Yeah, but
it's a very stable
very, very stable. But you know, we are using now very potent antiviral agents, and some of them really have generated escape mutants, which are of big worry to us, because we are not sure whether the vaccines are going to protect them on the long run. And under antiviral agent, there are escape mutants. So what we are seeing today is not necessarily what we are going to see tomorrow.
One more question?
Thank you.